Okay, so first of all, I would like to thank the organizers for the invitation. I'm very glad to have the opportunity to talk today about one of my favorite topics, which is bistability and bistability detection in biochemical reaction networks. I would like to start by introducing our group, because we have just started recently this last November in the Institute for Integrative Systems Biology in Valencia. Our group is the Computational Synthetic Biology group, where we work on biomolecular networks with relevance in cell regulation, signaling, and metabolic processes. And we are interested in applications for systems and synthetic biology, but also to advance fundamental knowledge and understanding on these systems. So biomolecular networks is how we call sometimes biochemical reaction networks in the context of systems and synthetic biology. And for these two disciplines, biomolecular networks are central because they are the models, the mathematical formalism that we use to describe metabolic pathways, signaling pathways, and gene regulatory networks. So in the group, we work on the design, the optimization, the analysis of biochemical reaction networks, both in deterministic and stochastic regimes, and also in modeling and control of metabolic pathways for applications in industry, like, for example, bioproduction in cell factories or fermentation processes in relevant to food industry. Today, I'm going to concentrate on the analysis of biomolecular networks, and specifically in the general problem we are interested in is establishing mappings between mechanism and function. So the topology, starting from the topology and parameters of a biochemical reaction network, being able to assess the behavior of the system, and if cells endowed with these biochemical networks or regulatory networks are capable for cellular decisions, oscillations, pattern formations, any functionality of interest in biology. And in particularly, I'm very interested, and we are very interested in the property of bistability, because bistability is related to the capacity of cells to take reliable decisions. So changing from one phenotype to other phenotype in a robust and reliable manner. And how is this? How bistability is related to this capacity? So if we have a bistable biomolecular network, like, for example, this signaling pathway in the plot, if we plot the dose response curve of this bistable system, so we plot the steady state of the species that we measure as a readout versus the input, and we increase gradually the input, the system will switch at some point from one phenotype to other. So we see an abrupt jump from one phenotype to other for a given value of the signal, for a given threshold. And if we do the reverse, the backward experiment, starting in the on state or this on phenotype, and we decrease the signal, the system will go back to the previous phenotype, but for a different threshold of the signal. So basically, this is what we call hysteresis, and this is the property that ensures the reliability of the decision of the change of phenotype. Because if we are in the on phenotype, the system will not go back immediately to the off phenotype, even if we remove the signal. So we have this margin of safety for the cell decision. That's why we say that it's reliable. So this type of dose-response behavior, which is the dose-response behavior we observe in bistable system, where we find this hysteresis, is responsible for these robust and reliable cell decisions. And this dose-response behavior corresponds to this underlying bifurcation diagram of our system, where we have a bistability region that is enclosed by two fold bifurcations that also in the literature are denoted as limit point bifurcations or saddle node bifurcations. So in this bifurcation diagram, within the range of bistability, we have two stable equilibrium branches and one branch of unstable equilibria. And the system will go to one steady state or to other steady states, depending on the initial condition. As you can see in this phase space plot, where we plot the trajectories for different initial conditions, and the system will end up in one steady state or other steady state that correspond to the different phenotypes. So these bistability-based cell decision-making mechanisms are ubiquitous in nature. So we find it from a virus in the lysis-lysogeny switch to the cell differentiation in mammalian organisms. And bistability is getting also more and more important to explain the molecular basis of disease in many examples, where the different states of cells, including disease and normal states, correspond to the different stable steady states of the underlying gene regulatory networks, and the transition from one state to other is understood or is represented. It happens as a bistable switch. This is a nice example, for example, that you can find in this paper in cancer research. So bistability detection is a very relevant problem in biology. And it's also a challenging problem because our networks, our models, our systems are very complex, and we also have a lot of uncertainty in the parameters and also even in the topologies. So we cannot directly use standard bifurcation tools to detect bistability in this type of systems without a lot of prior information. So a long time ago, back in 2003, when I started my PhD, our goal was, okay, we want to tackle this problem of bistability detection to be able to assess, starting from the graph of a biochemical reaction network, whether we can find bistability in a general and in a systematic way. So at the time, we found the lectures on chemical reaction network theory by Feinberg that you all know. And there we found the mathematical structure and formalism that we could exploit to derive a parametric condition for a limit point in kinetic models. And this parametric condition is central for our methods for bistability detection, because it exploits the structure of chemical reaction networks, because we derive directly from the graph, from the network matrices, and directly from the graph, and it's an exact algebraic expression. So it's very easily to check computationally. It's an expression in terms of the parameters, and then we can easily search through parameter spaces to find if this condition is fulfilled for which parameters. So what is the relationship between limit points, multistationarity, and bistability? So if this is the ODE system, the ODE model describing our biochemical reaction network, X is the state vector and B is the bifurcation parameter. M is the equilibrium manifold that you can see here plotted in the bifurcation diagram. So this is the locus of equilibria in the extended state parameter space. A limit point is a point such that is a regular point. So if we compute the extended Jacobian computed with the Jacobian with respect to the states and the Jacobian with respect to the bifurcation parameter, this Jacobian is full rank. So this is conditioned for a regular point. And the vector in the kernel of this matrix Q, at the limit point, the last component of the vector is 0. So the tangent vector fulfills this at the limit point. So these are the conditions for a limit point. And if we have this additional second order condition fulfilled, then we have a limit point that is also a limit point bifurcation, because we have actual change of sign on this last component of the vector, which corresponds also to a change of sign of the eigenvalue, and then we with this second order condition we ensure that we are not in this other possible situation. So our strategy for bistability detection is based on, okay, we're starting from the network graph. We have derived these algebraic expressions for limit point conditions in terms of the parameters. And then this algebra, we can search very efficiently when these conditions are fulfilled in the parameter space, formulating the problem of searching for a limit point as an optimization problem. So at the minimum is where our condition is fulfilled. So we can very efficiently scan parameter spaces searching for parameter vectors that lead to a saddle node or limit point. And then once we have found the limit point, then yes, it's very easy to apply current standard bifurcation tools, so continuation algorithms. So starting from the limit point, we start forward and backward continuation to test if actually our limit point is a limit point bifurcation, and also if it leads to bistability or not. But we very efficiently find these limit points, and then automatically the bifurcation diagrams are computed starting from there. So the first condition that we have derived is for mass action kinetic networks and systems with mass conservation, where we know that trajectories evolve in a reaction simplex or reaction polyhedron. So the condition is basically a geometric condition because we know that at the limit point, the equilibrium manifold and this reaction simplex are tangent. So we could encode this through a matrix that is 0 at the limit point, a matrix in terms of the parameters, and a matrix that we can compute, as I said, directly from the graph. But what happens if we don't have mass conservation? Like, for example, in signaling, we have degradations of the proteins usually. So then we needed to find a way to tackle these systems. So for this, we have made use of a previous result from Krasvin and Feinberg, which was a condition for multistationarity in fully open biochemical reaction networks. And this is based not on the deficiency part of chemical reaction network theory as the other result, but in the injectivity-based approach. So what we did here, because the problem for us to use in signaling of this result, the limitation, I would say, is that it requires, this fully open requires that all the species are going in and out of the system, of the control volume. So we have arrows from and to the zero complex. And this, in signaling, it cannot be because we cannot have production of intermediate complexes, which are always appearing in signaling pathways. So what we did to tackle this is to extend this result, so use this formalism to prove a condition for a limit point in semi-diffusive networks. So basically, semi-diffusive networks are also open, but in a way that complies with the requirements or with the assumptions that we can make in signaling. So we don't have this type of reactions. So this is what we call semi-diffusive networks. So we also find this condition for semi-diffusive networks and mass action kinetics. And then we have implemented these conditions in software tools. So basically, the software tools are very practical to find bistability, because starting from a graph of any signaling pathway or gene regulatory network, just with a graph, the tool will compute symbolically the algebraic expression, search automatically through a parameter space using optimization algorithms, and start bifurcation diagrams from the limit points if there is a limit point in the system. There are limit points in the system. So the tools are available online. This is in MATLAB, and I would like also to comment on this and recommend this tool, which is more recent, because it's in Python. It was developed by Brandon Reyes from the group of Vladislav Petyuk in PNNL, the Pacific Northwest National Lab, and contains these results and also other results from chemical reaction network theory. So basically, these tools are very efficient. If the system is bistable, if the network is bistable, it will find a limit point in the order of seconds. So now I would like to show you an example of how these tools or how methods for detection of bistability are useful in real biological problems. And this is a nice example. This was a project in which the goal was to understand the differential activities of interferon. Interferon is a molecule that triggers binding to a receptor in the membrane of the cells, triggers the STAT signaling pathway, and then this leads to expression of some genes. And depending on the dose, we can find completely different responses. One is called apoptotic activity of the cell, that is related to the anti-tumor effect of interferon, and then is important to quantify this for the use of interferon in anti-cancer treatments. And the other is the antiviral response. And the question in the project was how coding through the same receptor and same signaling pathway, we have these completely different behaviors. So for us, the systems part of the project, our hypothesis was that, okay, we have a bistable system, there is a cell decision being made, and we want to find at which level is this decision made. So we partitioned the model of the cascade in different modules, different reaction networks, the reactions happening at the receptor level, on the JAK-STAT pathway, and also the JAK-STAT plus the feedbacks generated by gene expression. And to be honest, we assumed, we were thinking that we were finding bistability when we added these feedbacks. So applying these tools for bistability detection, we didn't find bistability at the receptor level, not at the JAK-STAT, and not even when we add the STAT1 feedbacks. Unsurprisingly for us, we found the bistability when we connected the two modules of the reactions at the receptor level and the JAK-STAT. And this result was very relevant because it means that the differential signaling and the cell decision is happening at the early stage signaling. And also, since also I told you that these tools compute automatically the bifurcation diagrams, we could explain through these bifurcation diagrams using as bifurcation parameter one parameter related to the stability of the ternary complex at the membrane, we could explain previous biological evidences and of how this stability of the ternary complex was essential for this differential signaling. So our result was actually explaining these previous biological evidences and how the decision was made and how the differential signaling was happening. So as a first conclusion, we have developed conditions, algorithms, and software for efficient bistability detection in biomolecular networks with mass action kinetics. But what happens, we have other kinetics, like is in many cases the case for gene regulation, that we have, for example, Hill kinetics or Michaelis-Menten in metabolic pathways and so on. So for this, we have proposed to directly compute numerically the Jacobians, and then solve the optimization problem to find directly the fold bifurcation. So we can encode actually these three conditions in an optimization problem, where this so the first the tangency condition is at the minimum of the objective function, and the other conditions are set up by means of constraints. And one, it can be seen as a drawback or as an advantage, is that we have to compute the Jacobians numerically. We do this by finite differences. And one advantage of this is that we avoid the symbolic computations. Of course, the other is, I like it much in terms of formalism, because we are searching for an exact algebraic expression. This is not the case for this numeric approximation, but avoiding these symbolic computations, we can handle really high dimensional ODE systems of more than 100 of equations on large parameter spaces. I mean, more than 100 parameters, loose bounds. And again, we have high computational efficiency. So if there is bistability, we find the limit point in terms of seconds. And we can use these tools not only for detection of bistability, we can use them for designing bistable biomolecular networks. We can start from a search space that is not only a space of real parameters, but we can also introduce integer variables in as decision variables. So we can explore simultaneously topology spaces, which are encoded by integer vectors, and parameter spaces using mixed integer nonlinear optimization. So basically the problem is similar, but now we use, instead of global optimization with real decision variables, here we use global mixed integer nonlinear optimization, where the decision variables can be real and integer. So we can start from a superstructure of a library of synthetic biological parts and find the topology and parameters leading to a bistable system. But not only that, we can actually set or try to find bistable systems with a target bifurcation behavior. For example, the location, the regions where we want the limit points are located. So we can start from a library of components in synthetic biology, so pieces of DNA, and find through optimization the biocircuit made of these parts, leading to a target bistable behavior. Or in what we call forward design, we can find motifs starting from superstructures, which motifs lead to a bistable target behavior that we want. And this is very flexible because we can, as target behavior, I mean we can impose that we want a bistable switch with a maximal bistability range, for example, and on and off levels as close as possible to targets. And then by this optimization algorithm, we will find the synthetic biology circuit or the topology and parameters that will lead to the behavior as close as possible to the target. And this is the approach that we have used in a recent, well, this recent paper is actually, we have actually submitted less than a month ago, where we use this to find mushroom bifurcations, synthetic circuits leading to mushroom bifurcations. Mushroom bifurcation is the name that is used to refer to this type of bifurcation diagram, where we find these 4 limit point bifurcations, and we have this characteristic mushroom shape. And these are very important systems as biosensors, because the on state is available only for intermediate values of the signal. So it has a very characteristic and very...
I'll have to ask you to wrap up in about a couple of minutes.
A couple of minutes?
Yeah, I'm sorry, it's a little bit...
Okay, okay, so then, yeah, I will just go to the last part, where we want to describe, what happens to bistability and cell decisions in presence of noise. So when we have in bacteria, gene regulation, gene expression is a stochastic phenomena. So in stochastic systems, when we have, so we have a biochemical network, but now in a stochastic regime, and when we have low copy numbers of mRNAs, the deterministic models cannot capture the behavior. And it's the chemical master equation, the equation that captures this behavior, but it's mathematically intractable, even computationally. So we have developed a very efficient approximation of the partial of the chemical master equation in terms of partial integral differential equation model that speed up a lot previous methods of approximation for this. We can find this in this toolbox. And thanks to this method, we could we know that in deterministic systems, cell decisions rely on this hysteresis and bistability, but what happens when we have noise? Experimentally, it was observed that we have hysteresis in very stochastic and noisy systems like bacteria, as you can see in this very famous paper of Lac-Operon. This was explained for stochastic systems in this way. So if the system started in a basin of attraction, it will remain there. Other basins of attraction will remain there. But if we start in the separatrix, the system will jump stochastically to one basin or another. But this is not compatible with the theory of stochastic systems, because chemical master equation has only unique stationary distribution for this type of systems, and so this is not compatible with dependency on the initial conditions. So using these PID models, we could demonstrate, actually, reproducing these experiments. We plot the dose response curves I'm finishing. We plot the dose response curves for this stochastic system, starting from different initial conditions, blue and red lines for the final time of the experiment. Here, this is the mean of the distribution at final time versus the input. So this is compatible with hysteresis, but if we let the system arrive into the stationary state, the hysteresis disappears. So we have demonstrated that hysteresis is transient in presence of noise, and this has strong implications in how cells make decisions in presence of noise. So basically, our future direction is to quantify these cell decisions in presence of noise and study bifurcation in stochastic systems and also other emergent properties. And just to finish, I would like to dedicate this talk to Antonio Alonso, who was really the person that his contribution was absolutely fundamental and essential for this work and for this line of work. And you can find here all his, most of his contributions to bioprocess engineering, and also specifically to kinetic systems and chemical reaction network theory. And then acknowledge, of course, to all the co-authors and the funding agencies and all of you for your attention. And I'm really happy to get your questions. Thank you.